You know, the difference between a B and an A star in Cambridge biology, it honestly often comes down to one tiny, easily avoidable mistake on syllabus section 1.2, viruses. We're jumping right into this explainer today because I really want to expose a critical error that students make year after year. It's the kind of error that can literally cost you an entire grade boundary before you even realize you've made it. Now, you might think you already know everything there is to know about non-cellular structures, right? Well, if you don't understand exactly how examiners are evaluating your answers, you are leaving some seriously easy marks on the table.
So let's expose this painful learner mistake right up front, before we even touch the biology itself. Picture this: you've spent hours revising, you've painstakingly drawn out the structures, you've memorized all the terminology, and you walk out of that exam room feeling absolutely confident. Yet, when results day arrives, you're sitting at a B instead of that A star you worked so incredibly hard for. It's frustrating, isn't it? So why does ordinary revision completely fail so many dedicated students when it comes to this specific topic? Well, it's what we call the B grade trap.
And here is a quote straight from the Cambridge Principal Examiner reports that brilliantly exposes this trap. They say, "The classic mistake is providing generic answers not specific to the question context." So what does this actually mean for us? It means you can write down 10 perfectly accurate biological facts about viruses, but if those facts aren't tied directly to the specific experimental scenario or case study in the question prompt, well, you get 0 marks. Ouch. Examiners are looking for context, not just a data dump of everything you've memorized. All right, moving into section 1. We're looking at core architecture, nucleic acid core, and capsid.
What's really interesting here is how simple the definition actually is. A virus is a non-cellular structure consisting fundamentally of a nucleic acid core and a protein capsid. That's literally it. Understanding this non-cellular architecture is going to be your first quick win. Think about it. Because they are non-cellular, they completely lack the organelles we'd normally see in eukaryotes or prokaryotes. Keeping this structured reasoning front and center is a game changer because it totally prevents you from accidentally referencing cellular processes when you're analyzing viral behavior in a tricky exam question.
Now, in just a moment, I'm going to show you the examiner clue that changes the answer completely. But first, you kind of need the framework that makes that clue super obvious. Cambridge actually assesses your knowledge of that viral structure across 3 distinct cognitive domains. So you don't just need to know the facts. You also need to know how to handle complex data about those facts, and you have to evaluate experimental skills related to them. It's the whole package. To conquer those domains, here is our mastery framework for today's explainer. First, core architecture. Second, the method mark strategy. Third, flawed versus perfect application. Fourth, experimental skills. And finally, a final recap. Let's get to it.
Okay, section 2, the method mark strategy, process over memory.
When you hit those unfamiliar, highly specific contexts, like a totally new viral strain on the exam, you absolutely must use this specific problem-solving sequence to secure every single available method mark. First things first, you deconstruct the given information. Then, you solve systematically. And finally, you evaluate and recommend based on the actual evidence.
So here's the crucial point. You literally cannot skip straight to the answer. Top-scoring candidates always identify the key elements of the nucleic acid core and capsid mentioned in the prompt first. They break their analysis down into logical steps, they show super clear working, especially in calculations, and they are constantly checking the reasoning. Remember, you are proving to the examiner exactly how you think, not just spitting out what you know.
This kind of systematic processing, it's heavily tested in the statistical analysis of viruses. For instance, if a question throws data at you regarding viral mutation rates, you have to select the exact right statistical test. So you'd use the t-test for comparing means, chi-squared for categorical data, and correlation test for spotting relationships between variables. If you choose the wrong test, it immediately shows the examiner that you aren't actually evaluating the context.
But before we apply this to a real exam scenario, you really need to know the top 3 warnings pulled directly from the Cambridge Principal Examiner reports. Honestly, these are the exact traps that consistently tank even the highest performing students.
And here they are. Mistake number 1, forgetting units and calculations. I mean, just imagine calculating the replication rate of a virus perfectly, showing all your work and then losing the mark simply because you forgot to write down the units. It hurts just thinking about it. Mistake number 2 is not checking your terminology, like accidentally confusing the protein capsid with a cell wall, for example. And mistake 3, as we've already highlighted, is writing answers that completely lack context.
So, to really lock in that terminology for the nucleic acid core and capsid, you should craft a personal memory hack. Seriously, take a quick moment after this explainer to create a mnemonic using the first letters of key viral terms. The crazier and more personal the phrase, the better it's going to stick in your brain when you're under that intense exam pressure. That's a surefire way to make sure you never drop marks on mistake number 2.
All right, moving on to section 3, the application, flawed versus perfect answer.
Let's look at a flawed answer first. It usually reads straight out of a textbook, like, the virus has a nucleic acid core and a protein capsid. Now that's completely true, right? But it's also incredibly generic. Now consider the perfect, top-band answer. It takes those exact same structure facts, but explicitly links them to the scenario data. It sounds more like, because the scenario details a retrovirus, the nucleic acid core contains RNA rather than DNA, and the specific glycoproteins on its protein capsid allow it to bind to the host cell receptors. See the difference?
Okay, let's test your eye as an examiner for a second. Can you spot the error in standard generic answers? Often, questions are going to ask you to compare a virus to a bacteria. A major requirement for comparative analysis is identifying similarities and differences, while obviously supporting each with evidence. This means you absolutely must use comparative connectives, words like whereas, however, or conversely. These words actively signal to the examiner that you are evaluating the data, not just blindly listing separate facts in parallel.
The thing is, top-performing students consistently exhibit this kind of evaluative thinking. They question assumptions presented in the data, they consider different biological perspectives, and above all else, they justify every single decision with hard evidence from the text or the graph. It's this connected, conceptual understanding that easily transfers over to those really difficult, unfamiliar exam contexts.
On to Section 4, Escalation, Practical Experimental Skills. Now, if you're ever asked to design a practical investigation, you must write out your method following this exact sequential framework. Please don't just guess your way through it. Explicitly state how you will select the appropriate apparatus. Detail those step-by-step instructions. Explain exactly how you're going to observe and record the data in suitable tables. Make sure to mention how you'll identify anomalies in your viral samples. And finally, how you'll draw an evidence-based conclusion. It is literally the method mark strategy in action.
And this really brings us to a golden rule straight from the syllabus itself. Making cross-topic connections across the Biology 9700 syllabus, that is the ultimate secret to unlocking that A-star boundary. When you can seamlessly connect the non-cellular structure of a virus to other topics like immunology, cell division, or even genetic mutation, you are demonstrating a holistic mastery. And let me tell you, that's exactly what examiners are desperately looking to reward. And finally, Section 5, the final recap and the missing piece. Let's wrap this up.
Definitely remember these 3 core takeaways when you're practicing past paper questions later. Number 1, context is mandatory. Just promise yourself you'll never write a generic answer again. Number 2, use that deconstruct, solve, evaluate method to secure every single one of your process marks. And number 3, always include your units and double-check your terminology.
But wait a second. You could execute the method mark strategy perfectly. You could link every single structural detail of the capsid directly to the scenario. Your calculation units could be absolutely flawless. And yet, there is still one fatal trap that overrides everything we just learned today. It is honestly a silent killer of grades.
For example, you could be just describing when the examiner actually wants you to explain. It's a huge difference. So, to ensure a misunderstood command word doesn't completely destroy all your hard work, hit subscribe to ace the exams and jump right into our next explainer. We're going to uncover the command word secrets that literally decide your final mark band. See you there.